activation of transcription is a very important process during the protein synthesis, during the cell regulatory mechanism. And in this segment, we will study the transcriptional activation role of different factors like enhancers, promoters, and the coactivators. What are their roles in the transcription activation of a cell process? Now, the promoter region actually is a region of DNA that initiates the transcription of a particular gene. A particular gene is initiated, its transcription is initiated by a region of DNA which is known as promoter. And the promoters, as we know, they are located near the transcription start site of a gene on the same strand, and they are upstream on the DNA forwards toward the 5' region of the sense strand. That is the 5' region जो है उसके ऊपर ये promoters जो हैं they are situated, and the promoters are, as a matter of fact, those segments which initiates the gene transcription, कि gene को वो transcript करें. For example, here we can see in the DNA there is a activation protein, there are enhancer, there are promoters, and there are initiator, interceptor from one way to another to the upstream, we will call it, toward the 5' site of the DNA. Now, PEP-CK gene promotion or promoter, for example, let's study this. Now, in this case, the glucocorticoid response element, that is the GRE, that is a simulated or situated upstream from the PEP-CK gene, and they are generally considered to be the part of the promoter. They are referred to as a distal promoter, the promoter which are far away from different at a distance, and the elements to distinguish them from the proximal element which are situated closer to the gene. So one is situated at the close to the gene, and the another one, they are situated at the distal end, at the far away end of the gene. For example, here it has been demonstrated how the activation of a gene by a steroid hormone such as cortisol that takes place. And as we see that in the stage 1, that hormone, it passes down into the cell, goes down, then in the stage 2, it enters into the cell where it goes further from the stage 4 and 5. It goes to the act as a transcriptional factor and bind to a glucocorticoid response element, that is the GRE of a DNA, and eventually in the stage 6, it goes out and controls the transcription of this hormone. Now, the enhancer, that is the expression of most genes, that is also regulated by even more distant DNA elements, that is the elements which are far from the distant, and they are called enhancer. They activate the transcription. And an enhancer typically extends about 200 bases pair in length and contains multiple binding sites. बहुत ज़्यादा binding sites इसमें हैं for sequence specific transcriptional activation or activators which are there. Now, deletion of an enhancer can decrease the level of transcription by 100-fold or more. So, if an enhancer is deleted from this process, then it decreases the level of transcription by 100-fold or more because there is no more enhancer present. Enhancer and promoters, they may be separated by a large number of nucleotides. इकट्ठे नहीं हैं, they can be separated. However, enhancers are thought to stimulate transcription by influencing events that occur at the core promoter. ये इनका काम है. Now, for example, here we can see a assembly of basic transcription complex where the enhancer and promoter, they have been shown in different colors, and it has been indicated that how they act and take part in the enhancement of the transcription control. Now, another factor, which is the insulator, that can interact with the promoter over such long distances, and that is to prevent an enhancer from binding to an inappropriate promoter. जो insulator हैं, ये long distance पे उसके साथ act करते हैं, and their function is actually to prevent an enhancer from binding to an inappropriate जो है promoter उसके साथ. It is called an insulator, जिस तरह आम insulator होते हैं. Now, in this case, the transcriptional activator module or the core promoter has been shown, where there is an accelerator transcription, that is the enhancer, coactivator, and in the middle, that is the basal structure of a simple promoter, which is there, and how it controls the transcription level that's shown. Now, the coactivators, they are the mechanism by which transcriptional activator, they bind to an enhancer and is able to stimulate the initiation of transcription at the core promotion. So the coactivator, these are the mechanism by which transcriptional activator, they bind to the enhancer. तो इसमें क्या है कि transcriptional activator, it bind to the enhancer and is able to stimulate the initiation of transcription at the core promoter. And transcriptional factor which are accomplish this feat, which accomplish this through the action of intermediary, that is known as the coactivator. So in this chapter we have discussed the various factors, various things which are, as a matter of fact, responsible for a transcriptional activation by the cell.